Asalamo Alaikum, everyone. This video is going to be the last chapter exercise solution. So let's start. 1. Multiple choice questions, MCQs. 1. Which of the following is not a function of proteins in the body? Answer. B. Storing genetic information. Note, genetic information is stored in nucleic acids, DNA/RNA, not proteins. 2. Which of the following best describes proteins? Answer. B. Polymers of amino acids. 3. Which bond is responsible for linking amino acids together in a protein? Answer. Peptide bond, a covalent bond formed between the amino group of one amino acid and the carboxyl group of another. If you must pick one option, label peptide bond. A peptide bond is a specific covalent bond. 4. Which of the following is a component of nucleic acids? Answer. C. Phosphate group. 5. Which of the following is a characteristic of vitamins? Answer. B. They're essential for normal body function. 6. Which vitamin is important for vision and immune function and is fat-soluble? Answer. C. Vitamin A. 7. Which acid carries genetic information? Answer. B. Nucleic acid. 8. Which vitamin helps the body absorb calcium and is fat-soluble? Answer. C. Vitamin D. 9. Which of the following is a primary function of lipids in the body? Answer. C. Long-term energy storage. X. How might a diet lacking in lipids affect the absorption of certain vitamins? Answer. C. It would decrease the absorption of fat-soluble vitamins, such as A, D, E, and K. 11. Which vitamin is essential for blood clotting? Important note, none of the listed choices, A, C, D, E, is correct. The vitamin essential for normal blood clotting is vitamin K. Answer. Vitamin K, not listed among the given options. 12. Which biochemistry application is used to determine parentage? Answer. D. Parental genetic testing, also called DNA fingerprinting/parentage testing. 2. Short questions. How does the body make protein? Proteins are made by translation of mRNA at ribosomes. DNA, transcription, mRNA. Ribosomes read mRNA codons. tRNAs bring specific amino acids. Peptide bonds form between amino acids to produce a polypeptide, which folds into a functional protein. What do vitamins do for our body? Vitamins act as cofactors or coenzymes, regulators of metabolic reactions, antioxidants, and are essential for growth, immune function, vision, bone health, and many biochemical pathways. They do not provide calories, but are required for normal physiology. How do parents protect their children from genetic disorders? Measures include genetic counseling before conception, carrier screening for known inherited disorders, prenatal testing, e.g., amniocentesis, CVS, when indicated, avoiding known environmental mutagens in pregnancy, and in some cases pre-implantation genetic diagnosis, PGD, with IVF to select embryos free of specific mutations. Differentiate between genetic engineering and gene therapy. Genetic engineering, deliberate modification of an organism's genome in plants, animals, microbes for new traits, e.g., GM crops. Gene therapy, medical approach to treat or cure genetic disease by adding, correcting, or silencing genes in a patient's cells. Gene therapy is therapeutic and patient-focused. Genetic engineering is broader and often uses germline or somatic changes in organisms. How is gene cloning different from therapeutic cloning? Gene cloning, copying a specific gene sequence to produce many identical copies for study or recombinant protein production. Therapeutic cloning, somatic cell nuclear transfer for stem cells, producing an embryo-like source of embryonic stem cells genetically identical to a patient to generate tissues for therapy, not creating a full organism. Goals differ. One copies genes, the other produces patient-specific cells/tissues. Explain why proteins are considered natural polyamides. Proteins are polymers formed by amino acids linked via amide, peptide bonds between carboxyl and amino groups. Because the repeating linkage is an amide bond, proteins are natural polyamides. How do pregnancy tests use biochemical principles to detect pregnancy? Home pregnancy tests detect the hormone human chorionic gonadotropin, HCG, in urine. The test uses antibodies specific for HCG in an immunoassay. When HCG binds, a labeled antibody produces a visible line or signal indicating pregnancy. Evaluate the role of nucleic acids in heredity and cell function. Nucleic acids, DNA and RNA, store genetic information, DNA, carry it across generations, and direct protein synthesis via mRNA, tRNA, or RNA. DNA provides the code. RNA mediates expression and regulation. Mutations in nucleic acids alter traits/disease risk. Nucleic acids also regulate gene expression and are involved in cellular processes, replication, transcription, translation, repair. 3. Long questions, full answers, clear exam ready. 1. What is the significance of nucleic acids as information-carrying molecules? Nucleic acids, DNA and RNA, are the molecular basis for heredity and biological information. Storage of genetic information. DNA's sequence of 4 bases, A, T, C, G, encodes instructions for building proteins and regulating cellular processes. Faithful replication. DNA replication allows genetic information to be copied and passed to daughter cells with high fidelity, with repair mechanisms to correct errors. Expression of information. DNA is transcribed to RNA. mRNA is translated into protein. This flow, DNA-RNA protein, is central to the central dogma. Regulation and adaptability. Non-coding RNAs and regulatory DNA sequences control when and how genes are expressed, allowing cells to respond to internal/external signals. Evolutionary change. Mutations in nucleic acids generate genetic diversity, enabling natural selection and evolution. Medical importance. Mutations cause inherited disease. Nucleic acid analysis underpins diagnostics, PCR. Sequencing, forensics, and therapies, gene therapy, mRNA vaccines. 2. Compare and contrast the functions and properties of lipids and carbohydrates. Carbohydrates. Structure, composed of C, H, O, general formula, CH2O, underscoring. Monosaccharides, disaccharides, polysaccharides. Functions, quick energy source, glucose. Short-term energy storage, glycogen in animals, starch in plants. Structural, cellulose in plants, chitin in arthropods. Precursors for other biomolecules. Solubility. Many are water-soluble, monosaccharides, and small oligosaccharides. Lipids. Structure, diverse. Triglycerides, phospholipids, steroids, largely hydrophobic due to long hydrocarbon chains. Functions, long-term energy storage, triglycerides, structural, membrane phospholipids and cholesterol. Insulation and protection, signaling molecules, steroid hormones, aid absorption of fat-soluble vitamins. Solubility, insoluble in water, soluble in organic solvents. Contrast, energy. Carbohydrates provide rapid energy. Lipids store more energy per gram for long-term use. Solubility. Carbs generally water-soluble. Lipids are not. Structural roles. Carbs give rigid structures, cellulose. Lipids form flexible membranes. Metabolic uses. Carbs are first line for ADP production. Lipids are mobilized during fasting. 3. Why is protein structure important? Protein function depends on structure at several levels. Primary, amino acid sequence. Secondary, alpha helix, beta sheet. Tertiary, 3D folding. And quaternary, multi-subunit assembly. Correct folding creates active sites, binding pockets, and structural frameworks, enabling enzymes to catalyze reactions, transport proteins to bind cargo, antibodies to recognize antigens, and structural proteins to provide support. Misfolding or mutation can abolish function or cause aggregation and disease, e.g., sickle cell, prion diseases, Alzheimer's. 4. Explain the significance of amino acids in living organisms. Amino acids are the building, Proteins. They provide structural units for enzymes, receptors, transporters, and structural proteins. Functional diversity through side chains, charged, polar, hydrophobic, enabling catalysis, binding, and structural stability. Precursors for other biomolecules, neurotransmitters, hormones, heme, nucleotides. Roles in metabolism. Some amino acids can be used for energy or converted into glucose or lipids under fasting conditions. Essential amino acids must be obtained from diet. Deficiency affects protein synthesis and growth. 5. Explain the process of cloning in biochemistry. Cloning can mean different things. Gene cloning. Insertion of a DNA fragment, gene, into a cloning vector, plasmid, followed by transformation into host cells, bacteria. The host replicates the plasmid, producing many copies of the gene. Protein expression can follow in expression vectors. Organismal cloning, SCNT, somatic cell nuclear transfer nucleus from somatic cell is transferred into an enucleated oocyte. Stimulation causes embryo formation. Can be implanted to produce a genetic copy. Reproductive cloning, or used to derive embryonic stem cells, therapeutic cloning. Steps. Gene cloning example isolate DNA, cut vector, and insert with restriction enzymes/ligase, or use Gibson/topo methods. Transform host select recombinants amplify and sequence/express the gene. Applications. Recombinant protein production, insulin, research, transgenic organisms, therapeutic protein manufacture, and regenerative medicine. 6. What is the difference between genetic engineering and gene therapy? Expanded answer. Goal. Genetic engineering modifies genomes of organisms to create desired traits. Agriculture, industry, research. Gene therapy aims to treat or cure diseases in patients by altering genes in their cells. Target. Genetic engineering often involves plants, microbes, animals, germline, or somatic. Gene therapy focuses on human somatic cells, usually non-heritable. Ethics/regulation. Gene therapy is highly regulated and clinical. Genetic engineering has broader commercial and ecological implications. Methods. Both may use recombinant DNA, viral vectors, and CRISPR, but their design, safety, and monitoring differ because gene therapy is clinical and patient-focused. 7. Why are vitamins essential to human health? Provide 2 examples with their functions. Vitamins are organic micronutrients required in small amounts for normal metabolism, growth, and physiological function. Many act as cofactors/coenzymes or are involved in signaling and antioxidant defense. Examples. Vitamin C, ascorbic acid water-soluble, needed for collagen synthesis, hydroxylation of proline/lysine, wound healing, and as an antioxidant, deficiency scurvy, bleeding gums, poor wound healing. Vitamin D, fat-soluble, regulates calcium and phosphate metabolism by promoting intestinal absorption of calcium, maintaining bone health. Deficiency rickets in children, osteomalacia in adults. 8. Compare and contrast the uses and functions of proteins, carbohydrates, and lipids. Proteins, enzymes, catalysis, structural, collagen, keratin, transport, hemoglobin, signaling, hormone/receptors, immune, antibodies, movement, muscle proteins. Made of amino acids, diverse functions due to varied side chains and folding. Carbohydrates, energy, glucose, storage, glycogen, starch, structural, cellulose, recognition/signaling molecules, glycoproteins/glycolipids. Quickly mobilized for ADP production. Lipids, long-term energy storage, triglycerides, membrane structure, phospholipids, cholesterol, signaling, steroid hormones, insulation, and protection. High energy density, hydrophobic. Thanks for watching this series, and best of luck for your exams.